My name is Laura Figueroa. This is Chapter 4, describing the steps of protein synthesis. What is protein synthesis? Protein synthesis is a process in which cells make proteins. It occurs in two stages, which are transcription and translation. The three major types of RNA involved in protein synthesis are messenger RNA, also known as mRNA, transfer RNA, also known as tRNA, and ribosomal RNA, which is rRNA. Now that we know what protein synthesis is, let's discuss the steps that allow protein synthesis to happen. The first step we will be discussing is transcription. Transcription is a transfer of genetic instructions in DNA to mRNA. Transcription occurs in the nucleus of the cell. Transcription happens in three steps, which are initiation, elongation, and termination. Next, I will be going over the three phases that take place in transcription. The first phase is initiation, which is the beginning of transcription. Initiation takes place when the enzyme RNA polymerase binds to a promoter. This signals DNA to unwind so that the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. Elongation is the addition of nucleotides to the mRNA strand, and termination is the ending of transcription. The strand of mRNA is now complete and it detaches from DNA. After the strand of mRNA detaches from DNA, it must still go through a few more changes. The strand of new mRNA that was produced during transcription is not yet ready for translation. The new strand at this stage is called pre-mRNA, and it must go through more processing before it can leave the nucleus as a mature RNA. The processing may involve splicing, editing, and polyadenylation. Splicing removes introns from the protein coding sequence of mRNA. Introns need to be removed since they are regions that do not code for the protein. The remaining are regions called exons, which do code for the protein. Editing changes some of the nucleotides in mRNA, and polyadenylation adds a string of adenine bases or a tail to mRNA. It also signals the end of mRNA and protects mRNA from enzymes that might break it down. In this picture, we can see the pre-mRNA, which includes the exons and introns, and then the ribonucleoproteins remove the introns, and what is left is a mature mRNA, which consists of only exons. Now that the strand of mRNA has left the nucleus as a mature mRNA, the next step in protein synthesis will take place, and that is translation. Translation is the process in which the genetic code in mRNA is read to make a protein. Amino acids are linked together in a specific order according to the rules specified by the genetic code. Translation occurs in the cytoplasm, where the ribosomes are located. Ribosomes consist of rRNA and proteins. The ribosome reads the sequence of codons in mRNA, and molecules of tRNA bring amino acids to the ribosome in the correct sequence. Translation happens in four phases, which are activation, initiation, elongation, and termination. As with transcription, translation also happens in phases. Let's go over the phases of translation. The first phase is activation. So during this phase, the amino acid is covalently bonded to the tRNA. During initiation, transitional complex forms and tRNA brings first amino acid and polypeptide chain to bond to start codon in mRNA. During elongation, tRNA bring amino acids one by one to add to the polypeptide chain. And during termination, release factor recognizes the stop codon, the transitional complex dissociates, and the completed polypeptide is released. After the polypeptide chain has been synthesized, it may still go through additional processes. It may assume a folded shape due to interactions between its amino acids. It may also bind with other polypeptides or with different types of molecules such as lipids or carbohydrates. Many proteins travel to the Golgi apparatus within the cytoplasm to be modified for the specific job they will do. These are my references where I got the information for my video, besides my anatomy and physiology textbook. I also use these two websites where I got my pictures as well as information from. Thank you.